We finally know what it takes to be better at learning new information. Let's imagine a scenario. Suppose that you use a 6-digit code to unlock your mobile phone. For some reason, at some point, you need to change this code, and then you realize that it takes a while until you naturally enter the new code without repeating the previous one. Scientists have just discovered a brain circuit that promotes a more effective learning of this kind. How does the brain achieve this, and how can you benefit from it? That's what we are going to talk about today. The ability to adapt to novel situations is critical for our survival, and this ability is impaired in neuropsychiatric disorders as well as aging. But what determines our ability to adapt to a novel situation? Key to this fact is that novelty perception is encoded by several brain regions, including the ventral hippocampus and medial prefrontal cortex. Novelty was previously known to be involved in priming communication between the ventral hippocampus and the medial prefrontal cortex regions for subsequent learning by increasing the plasticity, a term used to describe neuronal flexibility of this brain circuit. Understanding how novelty primes brain circuitry to facilitate cognitive flexibility has importance not only for reversing the loss of learning capabilities during aging, but also to our daily lives in younger populations. If you have a strong pre-existing connection between the ventral hippocampus and the medial prefrontal cortex, you may be impaired in adapting to new tasks. The opposite stands true as well. If you disrupt pre-existing connectivity between the two regions, you enhance performance in cognitive tasks. The scientists behind the paper you can now see on your screen have hypothesized that weakening established circuit connectivity might increase learning capacity by permitting subsequent learning-associated plasticity. To test this, the authors have utilized a spatial learning task in which mice were trained to freely choose either arm of a T-shaped maze for food rewards. What happens in this task over time is that mice develop the most efficient strategy for choosing one arm consistently over the other. Following establishing their bias to a specific arm, the mice undergo additional training session termed flexible choice, which requires them to flexibly choose a different goal in the same maze. To successfully perform this new task, mice have to overcome their pre-established arm bias. While the mice do not fully overcome their bias, their performance gradually improves. And now to the role of novelty in this story. Following establishing a bias again in a new group of mice to investigate whether and how novelty affects learning, the scientists exposed the mice to a novel environment an hour before testing their behavior in the flexible choice training. As control for this experiment, a second group of mice were placed in a familiar context rather than in a novel environment. While initially both groups of mice kept entering the arm they were used to enter, mice that were exposed to the novel context progressively overcame this bias, improving in their performance more rapidly than mice exposed to the familiar context. In a lengthy series of experiments, the investigators verified that novelty activates the ventral hippocampus and that this process involves the neurotransmitter dopamine. Dopamine is released by neurons in another brain region called the ventral tegmental area, or VTA in short. The VTA is a brain region that is deeply involved in reward responses to both drug and natural compounds. In the context of the behavior described in this study, by releasing dopamine to the ventral hippocampus, the VTA provides information about novelty. In fact, directly blocking the receptor for dopamine in the ventral hippocampus prevents the effects of novelty on the efficacy of learning. We can't go here into all the technical details of how the researchers proved their point, but they were able to provide a solid line of evidence as to the mechanism by which novelty affects our ability to learn new procedures. In addition to physical exercise, one of the only physiological interventions that were shown to date to positively affect learning capabilities is environmental enrichment. The enriched environment paradigm exposes the organism to a variety of sensory stimuli that includes visual, auditory, olfactory, tactile, and social interaction, all of which contribute to the formation of dendritic spines which help form synapses in our brain. Theory suggests that the more synapses you have, the slower will be your cognitive decline during aging, since more synapses provide a greater cognitive reserve. This study extends this insight, and if we are to generalize these findings, we can say that one of the benefits for environmental enrichment is a greater cognitive flexibility, which is at the heart of neuronal plasticity and the ability to enhance memory.